Don't worry about a thing.
Hello, I'd like to welcome our watchers and listeners back to our new series called Making Waves, where we're doing an in-depth conversation with researchers that we're funding, supporting, that can shed some light on pediatric brain cancer and the complexities within it, but hopefully breaking that down in a way that people can make some sense out of. And I'm delighted to welcome Dr. Jason Cain with us today. Welcome, Jason.
Thanks, Liz. Thanks for having me.
Well, it's a delight to have you, and I've got your bio up. Hold on, let's see if I can. I'm just going to read this straight off of the Hudson Institute website, and you can elaborate on it. Jason is the head of the department in Cancer Biology Research Group in the Hudson Institute Center for Cancer Research, and you're a chief investigator for the Hudson Monash Pediatric Precision Medicine Program. Okay, well, that's quite a mouthful in and of itself. Would you mind giving our listeners and watchers just a little bit of your background and how you got to where you are now?
I'd love to. I'll try not to bore you with too many details, but so as you said, I'm a research group head, and the lab I run in the Centre for Cancer Research at the Hudson Institute is the Developmental and Cancer Biology Lab. So our main focus is on childhood solid tumours, and a large majority of that is in childhood brain cancer. So I got to this position through a bit of a journey. So I'm Melbourne born and bred, educated in Melbourne, went to university at Monash University. And my PhD was also at Monash, but it was actually in developmental biology, not in cancer biology, and in kidney development, of all things. And I undertook that with a really terrific mentor at Monash, Professor John Bertram. After my PhD, I wanted to undertake a postdoc overseas and was fortunate enough to get a position at the Hospital for Sick Children in Toronto, Canada. Which I loved. Amazing place, Toronto in Canada. And again, this was really in developmental biology. And my mentor, Dr. Norman Rosenblum, was actually a paediatric nephrologist at the hospital. So again, I was working with kidneys. But within my centre or department at Sick Kids, it was quite a diverse range of researchers and clinicians and research activities. And someone you know, Liz, Michael Taylor, had a lab in my centre, and this was mid 2000s, so 2006. And at that stage, for those who don't know, Michael Taylor is a paediatric neurosurgeon at Sick Kids in Toronto, and he was really just starting to gain momentum in his medulloblastoma program, along with his PhD student at the time, Paul Northcote. And so I was able to see the development and evolution of all of their research in medulloblastoma, which ultimately has revolutionized not only medulloblastoma, but pediatric brain tumor understanding of the fact that it's not just one tumor or one tumor type, but multiple molecular subtypes of the tumor. And really over the last 15 years, that has been one of the biggest developments in the field. And so at the time, I was so fascinated by this, and I thought to myself, I can't think, or I wasn't aware of anyone in Australia doing that kind of research and even work really in pediatric solid tumors. And so as I was looking to return home, I was fortuitously introduced to Professor Neil Watkins, who had just relocated to the Monash Institute of Medical Research, which is now called the Hudson. And he had come from Johns Hopkins. So he's an Australian who was at Johns Hopkins for a while and was returning back to Australia. Now, he was primarily interested in lung cancer. So that was kind of quite far removed from my expertise and probably where I wanted to go. But he had an amazing track record, coincidentally, in medulloblastoma. And so we got talking, and we had a lot of common interests in research. And he was really interested in helping develop a paediatric research program. So ultimately, when I returned to Australia, Neil and I developed the program that currently exists in my lab. And so that's kind of how I ended up to where I am now. So I consider myself a developmental biologist / cancer biologist. And I like to use developmental biology techniques to study cancer. And I think that's a really good fit for trying to understand childhood cancers.
Oh, great. And I'm gonna ask a few questions to hopefully help people understand sort of your role versus a clinical oncologist versus clinical trials. But before we get into that, obviously we're talking remotely, as everybody is these days, and hopefully we'll see some of that loosening here in Australia in the next couple of weeks, because Australia has done really well. But how have you found, now it's been several weeks, how's it been going for you, and what's happened with your work and what's happening within your lab?
Yeah, so I think, as with most institutes around the whole world, we're in shutdown at the moment. As you said, hopefully that will begin to loosen as of next week, in a staggered way. So essentially, all of my lab has been working from home. We have had some very limited access into the lab for essential work. So that is any ongoing animal experimentation, or really critical patient-derived cell lines that we need to maintain. Otherwise, they'd be lost. So we have been able to tick some things along, but essentially it's come to a little bit of a halt in the lab. But that hasn't necessarily been a bad thing, because this last 4 weeks or so has really given us some time to analyse data that we hadn't quite had the time to do, think about forward planning, about, what are our priorities moving forward? What are the really important experiments we need to do? Write papers, we've been doing that, and of course applying for grants. So we've been very, very busy at home, and as we're doing now, Liz, we've been keeping in contact with, my lab meetings continue. I continue to have individual meetings with my staff, and all of our really important collaborative research meetings with local and national and international collaborators have all continued. And so it's given everyone a little bit of time to pause and really think about what the priorities for us moving forward in the field are.
Well, that's good, and great to hear that things are progressing. And I think we're all adjusting. I call it they have business as usual. This is business unusual. We're all adapting as best as we can. Now, just because it's taken me, I've been involved for 7 years now, and it's been a massive learning curve for me, coming from outside of medicine, outside of research, really thrust into a whole new world. And I know many of our donors are similar. They believe in research. They think it's great that research happens, because we've seen so much success with breakthroughs in other diseases that only would have happened with research. But can you just, so starting at the beginning, so you're in more of what, people say basic research or discovery research. So the things you're trying to understand will then, you hope, lead to a clinical trial, which then you hope will lead to some new standard of care. I guess that's sort of a very simple way. But so can you just describe discovery research, and also how do you get an idea and then make it something that you actually want to explore in your lab? How does that process work?
That's a great question. So discovery research to me is really about understanding how something happens and why it happens. And so in the context of childhood cancer, it's what causes the disease in the first place, and then what causes it to continue to grow. And these are, they're really fundamental questions. But, I honestly believe that if you can if you can identify the reasons that these tumors start and understand why they continue to grow, then I think you're really well placed to devise strategies to stop those things happening. And so that would ultimately result in you hopefully finding a therapy that would be effective. So I think that's what discovery research means to me. And then obviously the next step is then to translate that research into the clinic in a clinical trial and hopefully an effective therapy. So how do we get at the questions that we address in the lab? First and foremost, we're in an area that we're extremely passionate about and we want to make a difference. So I think that's number one, because you have to be passionate about what you're doing because it can be difficult at times. And then number two, we ask what are the really important questions, and we get help in answering that because we have some terrific clinical collaborators. And at the Hudson, I have great relationship and access to the Monash Children's Cancer Centre, led by Dr. Peter Downey. So the Monash Children's Hospital is on the same side as the Hudson, so that makes things very easy. And, Peter and his team, they see patients every day. They know where the gaps in our knowledge and treatments are. And so that helps to frame the really important biological questions that we can ask. And so it's really in collaboration with the clinical staff that we come up with important questions. And then on the lab side, we design the experiments appropriately so we can answer those questions. And then once you kind of start doing that, it's the whole thing just keeps evolving. It keeps moving forward and evolving, and you acquire data, and then your next step and next question that you ask is then based on the data that you've just generated. So you can often move into very different directions based on different data points taking you in that way. And sometimes you can end up in places that you never expected to end up when you first started the project, but that's really cool.
And Jason, how do you know what if you're thinking about something here in Australia, is it sort of likely that your cohorts in Canada at the Sick Kids Hospital are thinking about it, at the DFKZ in Germany, at St. Jude's in Memphis in the US? So how do you know, do you work in isolation, or do you try to take a different tack than somebody else's, so you're trying to have a new perspective on a problem?
Yeah, so I think you don't want to replicate what somebody else is already doing. I think that's just a waste of everyone's time and resources, but it doesn't mean that you can't be involved in that project. And so I think paediatric neuro-oncology as a field is quite small, and there's many meetings that we have where we can interact with our colleagues overseas and we can hear what they're doing. There's really a lot of goodwill in this field, and people are all in it for the one reason, and that's to make a difference to clinical outcomes. So people are really happy to share what they're working on at the moment. There's not a lot of secrecy, and so you get to hear data that's hot off the press, so you kind of know what other labs are doing, and if you think that your work fits in with theirs, then you collaborate, and everyone is really happy to collaborate because we all understand that in such a small field, working on rare diseases where there are limited patients, limited resources, we actually need to work together in order to progress things as quickly as possible. And
How
so
does that work with IP though? Because isn't a lab's value or a researcher's value, and again, I'm new and trying to figure this part out, but isn't it sort of on the papers you publish and your names? How does that whole thing work? So is it sort of a balance between trying to collaborate, but also then trying to have an edge within your area, or what are your thoughts?
Yeah, no, so it is, it's a bit of a balancing act. So I think when it comes to publications, as I said before, there's so much goodwill in the field that people really want their data to be moving the field forward. So people are happy to collaborate and be on each other's papers. And if you can contribute to someone else's work, they very kindly acknowledge you in their publications, and vice versa. So I think, people are really happy to do that. And within this field, you often see papers. In fact, you rarely see papers where it's a single lab that's generated the data and published. It's just too difficult to do with the limited resources. And IP is an interesting one, because that's something we traditionally didn't really give a lot of thought to, but now labs are having to. But if there's something that's potentially, that has some value, commercial value in what you're doing, you can put, collaborative research agreements in place with your collaborators. So it's not prohibitive in collaborating. It just means that you might need to formalize the collaboration a little bit more carefully. But it's a really collaborative field, and as I said before, it's essential that it's like that in order to move things forward as rapidly as possible.
One of the things, and for the people listening, one of the recent things that we've begun funding at the Hudson Institute is a project. We've named it after a young boy who lost his life, whose family is involved with us, Alfie Chivers, and it's called the CRISPR project. Can you explain that in layman's terms?
Sure.
So what that technology is, and what the research we're funding, what we're hoping to get out of this?
Sure. So Alfie's project, the main goal of Alfie's project is to understand the drivers of childhood brain tumours, and atypical teratoid rhabdoid tumour, ATRT, is one of those that we're particularly interested in. And the main technique that we're utilising in this project, as you said, Liz, is the CRISPR-Cas9 technology. So this is a really interesting technology. It was kind of adapted from a bacterial immune defense system, and we can now utilize it in patient cells in the lab. And essentially it involves this protein called Cas9, but it's like this protein is like a pair of scissors. I think that's the best way to describe it. So it's like this. This is my Cas9 protein, and we can direct these scissors to different parts of the human genome, specifically to a specific gene that we're interested in. We can direct these scissors, and it will actually cut the DNA. It'll cut that particular gene, and then this cell will try and repair that DNA. But because we've made a cut, it's damaged a little bit of the DNA on either side of the cut. So when the DNA is rejoined, it's not exactly the same as what it was before. And because it's not the same, it therefore affects its function. So using the particular technologies we're using, we're actually making that cut, and when it rejoins, we're losing the function of that particular gene. And so we can interrogate any gene in the human genome, all genes in the human genome, at the same time in different cells, and determine how important that individual gene is for that particular cancer.
And is that what you were talking about when you were saying, really, what is causing the disease to begin with? Is this trying to get at that and digging deep into? So are you thinking that many of these pediatric brain tumors are genetic in nature versus environmental? So
some of them are genetic. They have, very specific mutations that are only seen in those particular tumors. Some of them are likely epigenetic. So that is kind of a change to the function of genes that's not moderated by a mutation. And so using these CRISPR technologies, what we can do is we can determine the importance of, as I said, any gene. And it may be that that gene is part of a important pathway that's been upregulated within that tumor. And that upregulated pathway may be the result of a a mutation downstream. But if we can find which genes are required for the growth of that tumor, then hopefully we can correlate that gene with a potential drug that's already developed. And if that drug is known to inhibit that gene that we can show is essential for the growth of the tumor, then very quickly we may have found a potential therapy to treat that particular tumor type. And really that's the goal of the project.
So this project, so the CRISPR project will identify these, is it targets within the gene?
Yeah, so we'll say, we like to call them dependencies, so genetic dependencies. So that means,
genetic dependencies.
yeah.
Once they're identified, then is that like phase 2, is to then try to find drugs that might be able to target them?
Yeah, so it's phase 2 in a sense, but we can actually do it simultaneously, because whilst we're doing a CRISPR screen on these cells, we can also do a drug screen on the cells. And so we can kind of meet at the top and then, match up the CRISPR screen with the drug screening data and show that when we delete this particular gene, we kill the cells. When we treat the cells with this particular drug, we kill the cells. And what do you know? This particular drug inhibits this particular gene. And so that's one way of doing it. Or we can follow it up in a phase 2 type approach, as you just said.
Okay, and this is interesting for us because 2 years ago, we were introduced at the Hudson Institute to a group that is, it was the Children's Brain Tumor Tissue Consortium. They've renamed or rebranded the Children's Brain Tumor Network. And for us, we were excited. I think many people know I'm American, you can hear it from my accent, but have lived in Australia for 20 years, but we have our foundation in both countries, which we're proud of. It's really celebrating Connor's legacy. He really was a boy of 2 countries. So when we met the people from the CBTN, we were excited because we liked to have that collaboration for us as a foundation between the 2 countries. And then obviously you've become quite involved with them. Can you describe how that relationship works?
So for those who don't know, the CBTTC or CBTN now, so Children's Brain Tumor Network, which is what they're about to be rebranded as, it's really a huge collaborative effort run out of the Children's Hospital of Philadelphia. So they've collected thousands of childhood brain tumor samples, and they've done an enormous amount of characterization of those samples, looking at the different mutations within each sample and various other analyses. And it's completely collaborative. So they give the tissues to other research labs, all the data they've generated to other research labs, and the purpose of it is to fast track individual researchers' projects with large amounts of data from rare tumors. So as I mentioned before, resources are limited. They're a network that collects all these resources together in large numbers and then provides them for researchers to undertake their projects. And so we've been involved for, as you said, Liz, at least a couple of years now. In fact, we're one of the member sites of CBTN, of which there's about 18 internationally at the moment, and that continues to grow. And we receive cell line models from CBTN with which to undertake our CRISPR analysis as part of Alfie's project. And along with those cell lines, we also get really important information about the cell lines. So which mutations these particular patients have, what gene expression patterns they have. And so all of that information is really important in correlating it with our own experimental CRISPR data.
So when you're working with them, so yes, you're using their data, but do they know what project you're undertaking? So then that in essence becomes collaborative, where they know what you're working on, and then another group might be working on another sort of angle. Is that how it works?
Yeah, absolutely. So they have a really streamlined application process to get their data and samples. And so that goes through a scientific committee that that coincidentally I'm a part of. Not that I would assess my own research projects, but we get to see, everyone on the committee gets to see what projects the network is supporting. And then there's regular meetings. So the scientific committee meets every 3 months. There's a PI meeting every month. And then they have an annual meeting where people who are undertaking CBTN collaborative projects will come and present their data. And this is, research in progress presentations. It's not necessarily finished projects. This is what we are doing and this is where we're up to. So it's a very transparent, collaborative process.
And Jason, obviously then the work, I've got 2 questions. One is the work they're doing and the work you're doing then would feed into clinical trials. And I know that they've got a strong alliance with PNOC, which is the Pediatric Neuro-Oncology.
Yeah, the Pacific Paediatric Neuro-Oncology Consortium.
Okay. So, and I know Australia is starting to become involved with that as well, which is another form of collaboration. And also from our foundation, we're funding work that's part of PNOC in the US, but now it's working from the Australia side as well.
Yeah, exactly. And so that's, I guess, if you look at the CBTN and PNOC now have a really terrific relationship because I guess CBTN is really facilitating the discovery research and PNOC is really driving that discovery research into clinical trials. And as you said, there's a number of partners in Australia with PNOC. And more recently, they've developed within PNOC these disease-specific working groups. And these are collaborative working groups on a specific brain tumour type. And these working groups have regular meetings. I'm part of the diffuse midline glioma working group. We meet every fortnight. That's incredible.
And I heard they've just started one for ependymoma as well, which is the tumor that Connor had. And again, for our listeners, this is super complex. There are, from last count, over 140 subtypes of different tumor types, which I think can be sort of bucketed into maybe 5 or 6 sort of main types. But then as you were talking originally about how you can classify them into these subgroups, it starts becoming really complicated.
Absolutely. And the beauty of these PNOC working groups is that with regular meetings, it's people presenting their data as it comes to hand. And so this is a really brilliant example of how collaborative research can progress into a clinical trial. And it's really aimed, these working groups are really aimed at collaborative work. So instead of 5 labs doing the same thing, you know exactly what everyone's doing and you say, hey, I can actually do this. This is my expertise. How about I do this? And that will complement what you're doing. And together we're going to get to the end game much quicker.
Jason, there's a lot of talk about precision medicine. You hear it not just within pediatric brain cancer, but you hear it with anybody who's getting a disease. Can you just very simply let people know what is precision medicine and how is it working in the pediatric brain cancer space?
Sure. So basically precision or personalized medicine is adapting a treatment to that specific individual. So instead of someone diagnosed with a particular brain tumor coming in and receiving just normal, just everyday treatment that may or may not work, that particular patient's tumor is studied, and we try and work out, or it's established what the unique, genetic mutations in that tumor might be, and trying to match those unique features of that patient's tumor to a therapy that is most likely going to work in that patient. And so the idea of that is that patients will receive, firstly, treatments that are likely to benefit the patient, and also hopefully not receiving treatments that are not likely to do anything but might have side effects. So that's really the brief explanation of that. Now, I mean, there's lots of ways that this is evolving in childhood brain tumors at the moment. I think one of the key features, one of the most obvious ways at the moment is the AIM brain study, which you know a lot about, Liz. And this is the methylation profiling of every childhood brain tumor in Australia and New Zealand. And that information tells the clinicians the exact molecular subtype of that particular tumor. And as you said, Liz, there's all these different types of brain tumors, and within these different types of brain tumors, there's all these different types of molecular subtypes. And that means that those subtypes are driven by different genetic mutations or different activation of pathways. And therefore, a specific subtype might benefit better from one therapy versus another subtype, even though they're under the umbrella of the same disease. And I think that's the best example of how precision medicine has evolved over the last kind of 18 months to 2 years in that space. And then it's been refined more and more and more as we move along.
Well, we were that was, in terms of my proudest moment, was when we were able to really get behind the AIM Brain project and be a primary funder and really help bring that to fruition, because it really is sort of a base for then all this other research and everything to stem from there. Now, Jason, we just have another minute or so, and I'd like to end these little interviews, these in-depth, and you've been so informative. I hope people have enjoyed it as much as I have. I just wanted to leave with, what are your greatest hopes for, when you see, I know you work with Dr. Peter Downey, and he's in the front line dealing with the patients and the parents, but from your perspective, you said you're so passionate. So what is it that makes you passionate, and what are you hopeful for?
What makes me passionate? I have kids of my own, and it kills me to think that parents and families and children have to go through what they have to go through. And it's terrible that, we haven't advanced this area in pediatric brain tumor therapies as much as other cancer fields have been advanced. So, my hope is that moving, in the future, that all patients have access to curative therapies that have no side effects. I think that's the ultimate goal that we're all in this field working as hard as we can towards. And I think that is the ideal, situation that we want to find ourselves in.
Well, I couldn't agree more. And I know people listening will find it also so inspiring to know that there's people like you trying to crack this code and figure out how we can help these kids with such a cruel disease, as you know. Jason, thank you so much. We're very proud to fund the work that you and your group are doing, and we hope we can continue that relationship for many, many years to come.
Thank you, Liz, and thanks to the RCD for all your support.
Thank you. Have a good day.
Thank you.
Take care. Bye.
Bye.
'Cause every little thing is gonna be alright.